Okay, we're into part 2 of the session on oncogenic mutational profiling of tumours, and what I'm now going to focus on is how the mutations are determined by whole genome sequencing or whole exome sequencing, and I'm now going to cover the sequencing methods. So there are two old methods that I will briefly cover just so you understand where sequencing technology has come from, and this is old style Sanger sequencing and what we call the DigDye Terminator technology. And Sanger, old style Sanger sequencing requires use of radioactive phosphorus or radioactive sulfur, depending on how we're doing it, and requires the use of what we call dideoxy chain terminator nucleotides. And these chain terminator nucleotides, effectively, what you do is have a piece of DNA, allow DNA polymerase to copy it, and put in some chain terminator nucleotides in with the regular nucleotides, and every now and again the DNA polymerase will stop because it's incorporated a chain terminator that can't be extended. And wherever a piece of DNA stops, you then get a set length of DNA, and if you include dideoxy, which is the chain terminator, A into one tube and G and C and T into the other tubes, you can see where these reactions stop, and that gives you effectively a length of DNA, and I'll explain this in more detail on the next slide. And then we go on to the DigDye Terminator technology. This is using fluorescent chain terminators, so it's the same idea as this. You have to run your sequencing reactions on 4 wells of a big long sequencing gel, and then perform autoradiography of that, whereas here you run them all down effectively a column, and you get a red, green, blue or yellow trace depending on which colour nucleotide is last incorporated onto the sequence. So this sort of explains how the dideoxy chain termination sequencing works. We start our DNA replication on a piece of DNA. We'd supply a test tube with a piece of DNA that we wanted to sequence. We have a primer. That's a short DNA strand that creates a double-stranded primer, a double-stranded region of DNA for DNA polymerase to start from. That would have a 3' OH group attached, and DNA polymerase will be able to extend from that. So this would be the primer on a template strand. We supply the enzyme with deoxy A, G, C and T, and we also incorporate, we split the reaction into 4, and into one reaction we incorporate about 10% dideoxy ATP, then 10% dideoxy CTP, GTP and TTP in 4 separate reactions. We allow those reactions to go separately and then run them down a big long sequencing gel. And effectively what happens is wherever you've incorporated, from extension from this primer, where you've incorporated a dideoxy T, the DNA sequence will stop at this point, and the DNA length will be from here to here, and that is that one. And if you then incorporate a dideoxy A and a different reaction in the other tube, the DNA polymerase will get to this point and then stop, and that's given this length fragment and this length fragment. Remember, we're incorporating having about 10% dideoxy, 90% normal nucleotide, so we don't stop at every time DNA polymerase tries to put in, let's say, a T. It will stop in a small proportion of cases, and we basically run the sample down the gel, 4 different reactions down 4 different wells, and we read the sequence TAA GGA GGT, and that tells you the sequence along here. And that is old style DNA sequencing, very inefficient. We then move on to the DigDye Terminator, much more automated, much more high throughput, and in this situation we have a template. Here's the primer binds to the template, DNA polymerase extends in this way and incorporates either a dideoxy TTP in red or a dideoxy CTP in blue. And we allow the DNA polymerase to go as far as it can before it encounters a chain terminator, just as in the previous example. The difference here is all of these are in the same tube, and we just run the whole lot down a column, do chromatography on this, and we see what colour each of the different size products are, and we get a chromatograph and we read the colours from shortest to longest. So if this is the short, if these are the first ones to come out, these are the last ones to come out, this is the start of the sequence, and we read the colours and convert that into DNA sequence. So this is mostly what was done up through, the Sanger sequencing was done through the 70s, 80s. I was doing that in the 90s. Then the DigDye Terminator through year 2000 onwards, and then we started doing whole genome sequencing using one of two technologies. I'm going to teach you about the Illumina platform. Okay, so at this point it's probably a good idea to click on this link and read the Illumina document. It tells you how the Illumina sequencing platform works. Then click on this link and watch the Illumina sequencing technology video. Now the problem is, if you watch this video first, it will make no sense. If you read this first, and I'll go through the key points of it first, then go back and watch the video, and the video might make sense. Otherwise, the first 30 seconds of it will make no sense. So what I'm going to do is go through effectively the key points of this document first, and then you can watch the video. So this is a quick summary of how the sequencing works. Firstly, we get our DNA that we want to sequence and we fragment it. And then it's fragmented, and then we add on one of two different linker sequences onto the end. These are oligonucleotides. They're short pieces of DNA, and we stick one on one end and one on the other. So we are randomly fragmenting the DNA and then sticking these linker molecules onto the end. These linker molecules are actually linker oligos. They facilitate what we call isothermal amplification on a solid matrix. So that means that we're going to attach these oligos, and these oligos are going to be captured by a solid matrix which have complementary oligos that match the sequence. So we've got millions of pieces of DNA, we've tagged them on the end with a linker oligo, and then that linker oligo is going to be captured onto a glass slide which is coated in a lawn of oligos which match one of the two that we've tagged onto the piece of DNA that we are interested in. That allows us to perform isothermal bridge amplification, which I'm going to talk you through. So we perform that is something very similar to polymerase chain reaction, but it's solid phase and it's on a chip, and I will talk you through that. But that will create what we call local amplification clusters, which is a cluster is made of one piece of DNA which landed on the chip and then got locally amplified in a very tiny area. So you've got thousands of copies of this piece of DNA all in the same localized area on the glass chip. What we then do is use primers to one of the linkers that we attached in the first place, and then perform sequencing by sequentially adding in nucleotides of different colours and seeing which ones fluoresce when we excite them with a laser. So if we can focus on one little patch of the glass slide which has got a cluster of DNA that's been amplified there, we can add the nucleotide sequentially, excite with a laser, and see if when we added in T's we got a little red flash, and when we added in C's we got a little blue flash, and so on. We do this on a huge scale, and we're doing this simultaneously looking at millions of clusters on a single chip. Now each cluster should have the same sequence, so we're basically scanning for the emission of little flashes of light from different regions of the chip and following those over the course of adding in up to 100 nucleotides, and then keeping a record of that at each location and using that to build up a whole genome sequence. And that was at 0.5. So as each base is added, or each nucleotide is added, all the clusters in a flow cell that allow that next nucleotide to be added. So if the template is a T and we added in some A's, everywhere an A has just been added in, we'll get a little flash of light. Now this is a really clever bit. We fragmented our genome into about 100 base pair fragments, and all of the sequences that we get are in little tiny fragments, and we just collate all of those together and basically line them up and try and match them up. It's like doing the world's biggest but most impossible jigsaw, but we use supercomputing power to do this, and we align all of these millions and millions of sequences to build what is the most likely genome. So we've fragmented the genome, sequenced it, and then pieced it all back together using a computer. And these overlapping sequences give contiguous read, basically continuous read that will read effectively from the start of chromosome 1 right to the end of chromosome 1 and so on. And we can piece together an entire genome doing this. Now I would say don't watch the Illumina video just yet. I will explain these points in more detail. Okay, so here's our DNA, our genomic DNA. It's been fragmented and then it's had these adapters stuck onto the end. These are important for binding our DNA to a lawn of oligos on a glass chip. So here's the glass chip, here's the oligos that are already bound on there, and here is our DNA with the adapter oligos here and here, and this adapter oligo will bind to this one that is bound to the glass chip. So this basically tethers our DNA to the glass chip. This is the first step prior to doing isothermal amplification. The next step is bridge amplification. And what we want to happen here is this oligo that's been tethered here to fold over and meet another oligo that's tethered to the glass. So what you can see is this one here is tethered to this one, and it can fold over and meet another oligo that it matches, which is going to be nearby, and then we can copy one to the other. So this is our DNA that we want to sequence. This is tethered by one oligo here, one oligo here, and now we can add in DNA polymerase and it will copy this all the way around here till we get to this situation here. Now what's not shown is that this looks like it's stuck to the glass slide. It isn't, it's actually tethered by an oligo. But anyway, we can end up after this as having two pieces of DNA, one sense and one antisense, both attached to the glass slide. That gives us this situation here, and then we just repeat the process so that we end up with lots of copies of the same DNA in a local area, and we call that a cluster. And we can then do some other steps where we add in, where we cleave off one of these sequences, but we can start then adding in our nucleotides and primers and look for flashes of light when we add in a G, C, T or A on a localized area. Okay, now with that bit of basic knowledge, it's probably now a good idea to watch the Illumina video start to finish and keep referring back to what I've just said to try to understand what the video is doing so you're not immediately confused. Okay, so hopefully you've watched that video and hopefully you've seen how it works. For the purpose of this module, I will not expect you to be able to regurgitate and explain how that sequencing works. I think it's just useful to explain it to you to understand the scale of sequencing that is required to do a whole genome sequence, and it is on a vast scale. Now this slide shows you how the sequence is pieced back together. So what we have got is, imagine this is the whole sequence from start to finish for the genome. We have got lots of short reads, and each read is about 100 base pairs in length. And what we can do is overlap those reads, and in a typical whole genome sequencing experiment we might have got 100 million reads of 100 base pairs in length. And what we do is we just align them up and find out which ones align with each other. Now if there is an overlap of about 20 base pairs from here to here, well that sequence should only occur once in the human genome, so we can quite confidently say that sequence there and that sequence there are effectively overlapping with each other, and then this sequence here matches this little bit, so this must go next, and we just build up the whole sequence that way. And we try to get a read depth, if you get a read depth of 10, that means that each base has been read 10 times on 10 different sequences. So if we went up here, let's say whatever base that is there, it's been read once, twice, three, 4, 5 times. So that would be a read depth of 5 for that particular sequence. And the deeper the read depth it is, the more accurate the sequencing is likely to be. So that's what it's done. It is really, it's computer based. There's no way that we could do this any other way than just letting a computer take all those reads and piece it together as best that it can. All that happens then is that full-length whole genome sequence is compared against the reference genome for humans and look for anything that is different, and that gives us our somatic mutations in the cancer cells. So just to summarise all the different sorts of sequencing that is out there, and you'll see a lot of these in cancer biology. Only the old papers do Sanger P32 sequencing. You will not see that anymore. Sanger Big Dye, you maybe see that in some studies still. You might come across pyro sequencing. This is not ideal, but this is for very short reads. So if you've got a PCR product that you want to sequence, this is very good. Not for whole genomes. There's another sequencing system called Nanopore sequencing. Nanopore sequencing is interesting in that can read DNA as it passes through a pore on an artificial membrane. The beauty of this is you can do much longer continuous reads of hundreds of thousands of base pairs. So if you've got a situation where you want to read, you don't want to have to piece all of this DNA back together and risk mistakes being made. Nanopore sequencing can do that, and it can do long reads of hundreds of thousands of base pairs. It's the only technology that can do that. There's also Ion Torrent, which is also comparable to Illumina. It can do what we call, it's the same type of thing, massively parallel next generation sequencing, capable of doing whole genome sequencing. So those are the main sequencing technologies that are available, and this just summarises what they, when you would use them. So Illumina and Ion Torrent you can use for whole genome sequencing, whole exome sequencing. When you're looking for an unknown mutation in a genome, they're fine for that. Nanopore is more for if you want to read tens or hundreds of thousands of base pairs. If you want to, if you had a bacterial colony that you wanted to sequence start to finish, Nanopore could do that, but it wouldn't be able to do a whole genome sequence just yet. Pyro sequencing, a short read. It's sort of a replacement for the old Sanger deoxy method for a single PCR product. Big Dye, you can do a lot of sequencing, but nowhere near this level. An Ion Torrent or Illumina could do a whole genome in a day quite easily. This would take you weeks and weeks to do a single genome. Okay, that's as much as I'm going to say about the sequencing technologies. Again, the important thing is to understand what can be done with the sequencing rather than the actual technical details of how to do the sequencing. I've included it because I think it's really interesting. The important thing for you as cancer biologists is which tool can be used for which application. Now what I'm going to show you on part 3 of this session is where this sequencing data has been used and put into the COSMIC database. So I'm going to guide you around the COSMIC database so you can see for a single tumour type everything that is known about the genetics, and all of this has come from Illumina or Ion Torrent sequencing.